I'm Mathieu Dubernet. I'm CEO of Laboratoire Dubernet, which is one of the most important wine lab in Europe, private and independent. I'm going to talk to you about a new application of flow cytometry, which has been used in the next trials presented by Fermentis. Consider that from a clinical point of view, wine is no more than chemical parameters and biological parameters. Of course, it's not only that, but from a laboratory point of view, it is the case. During last decades, many improvements were done on the wine analysis on the chemical parameters, and these curves show you some of the most relevant and important innovation that allow the productivity of laboratory to enhance and offer today services that were not possible to imagine 40 years ago. But during this time, microbiology remains to the very old petri dish that is still the main way for wine analysis. Actually, it would be unfair to not speak about molecular biology that allow a lot of analysis, but molecular biology is still expensive and not always suitable for all the purpose, even if it's very well used here in California, but it's not the same in France, for instance. But wine microbiology have a lot of challenges to go with. First is Brettanomyces, the wine bottling, and we need to manage a good microbiology during all this process of bottling. The free SO2 wine, which is coming more and more in many places, the use of wild yeast or the use of dry yeast under quality control, it's very important. Actually, the fermentation monitoring, that is not allowed today by the routine system of analysis. And for all that is for reason enology in order to use fair process with the necessary and the sufficient processes. So flow cytometer is not brand new in the wine industry, but we made very new innovation. The principle of flow cytometer is to flow the wine through a very thin cannula where all the particula were counted by a laser beam and different optical treatment of this laser. Then you have this kind of system which is a cytogram. Each point is a particular in the wine, so could be bacteria, could be a yeast, or could be something else. And all the challenge in the cytometry is to make the distinction between the background noise and the relevant thing you want to actually measure, which are bacteria and yeast. And in yeast, to make the distinction, of course, between Saccharomyces and Brettanomyces, or other Enoforum, for instance. Cytometry has been considered for wine analysis a long time ago, because the very first publication are from the 80s. But since it has not been used a very large way because of the background noise, with limited performance, and actually today the technique still have a poor fame and is not used as much as it should. Principally because of the poor selectivity between Saccharomyces and Brettanomyces, it is very difficult with the classical way to make the distinction between both. But that was the old time. Now we have very interesting axis for the flow cytometry development. There are three of them. First is the use of fluorescence-activated cell sorting that are staining of cells that allow to sort them, and it is very, very efficient, and there is a large possibilities of staining strategies. Second is the use of molecular biology, actually, with antibody or antigen system with fluorescence. This is like a PCR. It is quite expensive. And the third way is the material improvement, which is very important during the last years. In my point of view, the one and the third way are the most effective to allow to have an interesting method that is low cost and very efficient. And this is what we did. The fluorescence-activated cell sorting is this way. This is under a fluorescence microscope, and you can see here Brettanomyces. And the stained cells, which are in green, are the alive, active Brettanomyces. The one in brown are the alive but latent Brettanomyces, which are not producing volatile phenols. It's very important to make the distinction between both and consider that PCR doesn't allow to make this distinction. The same thing, it's more difficult to see on bacteria, but it's very efficient as well. So thanks to the cell staining, it's possible to make the distinction between active living, which is the only one who grow on petri dishes, non-active living, and death. And basically, PCR allowed to see all together without a clear distinction. So the flow cytometer offers a very new, interesting way to make a relevant distinction between the physiological status of the different yeast and bacteria. Material improvement is thanks to very new systems that were not used still in wine analysis. They were used for biology or environmental analysis, with flow which are very, very fast, much faster than the old system, high sensitivity detector, and the use of different lasers. Actually, we are using 3 lasers and maybe 4 in the next generation. And we use a triple fluorescence staining for that purpose. And the application is very nice because it allows to make the distinction of Brettanomyces, Saccharomyces, bacteria, and it's possible to make the distinction between gram-negative and gram-positive, I mean lactic and acetic bacteria in wine. And the analysis lasts few seconds versus days and days for the petri dishes. And it's possible to consider application for all the follow-up of the winemaking, and including, of course, research and development. The question which is often asked is about Saccharomyces and Brettanomyces distinction, because nobody succeed to make this distinction before, and we can do this, and you can see here the cytogram. Unfortunately, I'm not going to tell you, oh, I do this today, because a patent is processing right now, and it's too early to tell you about. And the validation was being performed, large validation, because the method was accredited ISO 17025 in my laboratory, which is a high level of reconnaissance. It's not relevant to make comparison between petri dishes and flow cytometer. The reason is that it is never certain that one colony arose from a single cell. And many people make this mistake about the CFU interpretation. CFU is often 20, 50, or maybe more cells at the origin, depending on the stain of the yeast or the bacteria. So there is no correlation possible, but the cytometry gives you directly a reading of what you are in the wine. So then it's possible to have a routine system, low cost, very fast for to monitor all the microbiology. And first, of course, is the Brettanomyces. Consider that today in my laboratory, for instance, in France, we run any sample as well for chemico-physical parameter and as well for microbiology, and Brettanomyces are followed each month for any tank. So you have here the kind of results. And this is the rule of interpretation. We know that lower than 1,000, the Brettanomyces population of active living is not going to produce at short term volatile phenols, but over you have to react quite fast. The information that we have from this method that allows us to analyze thousands and thousands samples since 2 years. I mean, today maybe it's something like 100,000 samples of wine, is that Brettanomyces is everywhere. Brettanomyces should not be considered as a contamination yeast, is a yeast from the biodiversity of natural presence on the grapes than on wine. The question is not if you have or not Brettanomyces, you have, but the question is, my population is active or not, and if it is active, what is the size of the population? So you have to monitor Brettanomyces and consider that to have Brettanomyces in your wine is a normal thing. So the fight should be against volatile phenols and not against Brettanomyces, because against Brettanomyces the fight is lost already. It's a bit like the coronavirus, you know. Okay, so in one sentence, everything is said here. In white and rosé, it's about the same, but half of the wine we are analyzing doesn't have Brettanomyces anymore. The reason is that in white and rosé process of winemaking, the first steps remove a large part of the natural microbiology present, the guanine yeast present on the grapes, and they are not anymore. Second, on many of these wines, we have filtering and fining which are very at early stages. Then this method is very fast and easy to handle to check after the different process. For instance, here the use of chitosan, and you can see that it removes the alive active Brettanomyces, not the alive latent Brettanomyces, which is a very interesting information. For instance, here you have a treatment, and you can see in red that the curve of a live active doesn't reach the 0, and because the 4 grams per liter, which is the routine doses of chitosan, is not enough to treat all the population. And with 10 grams per liter, it's okay to remove in this case. So it's very interesting before a treatment to check what is my starting level of population, because any treatment in wine is going to lower your population, but not to remove your population. The same with flash pasteurization. It's okay to check after. We can see if your flash pasteurization is badly adjusted. You can monitor your tangential filtration, your bottling, and it's very interesting to make fermentation monitoring and check what is the status of your population. And we can see that you have very low population fermentation, and the fact is these are the slowest fermentation, and you have very fast, and the average you can see is about 2 to 7 of yeast per milliliter. But in fermentation, you have as well Brettanomyces, which are present in many, many cases, even if you don't have a production of volatile phenol. Yeah, and it is a very interesting thing to look into. And malolactic bacteria and malolactic fermentation is always nice to monitor, and you can see the population of bacteria before any malic consumption. So it's very interesting to check this way. Sampling is a new challenge to have the good sample for this analysis, but with the right method, it's not a problem anymore. I would like to thank you, and merci pour votre agréable accueil et votre aimable attention.